It is my great pleasure to introduce Dr. Delphine Othman to speak today. Dr. Othman is a postdoctoral fellow at the University of York. She's working on animal African trypanosome cell surface proteins to identify new vaccine targets with a special interest for Trypanosoma congolense and Trypanosoma vivax. Dr. Othman received her undergraduate degree in biochemistry, her MSc in physiology and neuroscience, humanitarian health degree, and a master in neuroscience from Université Claude Bernard in Lyon. She completed a PhD in cellular and biochemical sciences at the University of Bern in 2012, after which she was a postdoctoral research fellow at the Institut Pasteur in 2014. At the Pasteur, she investigated new therapies against Trypanosoma cruzi infection and tissue pathology. She was a postdoctoral research fellow at the Wellcome Sanger Institute in Hingston from 2015 to 2021, where her research focused on genome-led vaccine target discovery for animal African trypanosomiasis. She now continues this work at the University of York. She's been an active member of Aide au Développement en Biologie Médicale with Biologie Sans Frontières since 2007. I'm very excited about hearing about this work, the paper that came out, and I was really excited to read it, and I think it's going to be a great seminar. So over to you, Dr. Othman, and welcome.
So thank you very much for this nice introduction, and also to give me the opportunity to show our data regarding the investigation of vaccine targets against Trypanosoma vivax and Trypanosoma congolense, but today I will focus on Trypanosoma vivax. So animal African trypanosomiasis is really a major disease in sub-Saharan Africa. So this picture can really summarize the disease, because as you can see here, there is a really emaciated cattle, and around this farmer is really desesperated about it, because in certain region of Africa, the people really relied on their cattle, so for food first, but also for milk and draw power as well. And animal African trypanosomiasis have a very big repercussion on their livelihood. So to introduce the disease, so the disease induces chronic anemia with a rapid loss of weight, neurological dysfunction, muscle wastage, fertility and milk reduction, and death if the animal is not treated. So the disease can affect also cattle, but also water buffalo, sheep, goats, horses, pigs, dogs. So a lot of economically important animals, and also wild animals such as antelope, lions, elephants. So there is not a lot of animals that trypanosome can't infect. So the disease is caused by several species of trypanosomes, Trypanosoma congolense, Trypanosoma vivax, and to a lesser extent, Trypanosoma brucei. So as you can see here, the parasite has an elongated shape and has a flagellum all over his body, and is freely moving into the blood of the host, infected host. The parasites are transmitted by tsetse fly, but in the case of Trypanosoma vivax, not exclusively, because the parasite, Trypanosoma vivax parasite can also be transmitted by other flies such as tabanids. And that's probably the reason why the parasite has been exported from Africa to South America. So there is an estimation of 50 million cattle which are at risk of being infected by animal African trypanosomiasis, an approximately 3 million deaths of cattle every year. And this leads to a very major productivity losses every year. And in Africa, you really have areas where you can't grow cattle because it's impossible to keep the cattle in this area. So there is really area where it's impossible to have farmer activities. So at this time, the disease is managed using treatments. There is 2 curative trypanocidal treatments, which are indeed very old because this drug has been developed in the 50s, and now they are associated with increasing resistance. And there is no vaccines available. So to go a bit more into detail of the life cycle of Trypanosoma vivax, so the parasite has 2 different hosts. So a mammalian host and also the tsetse flies. So the cycle starts with the replication of the bloodstream form into the blood of the mammalian host. And then upon the blood meal of the tsetse flies, the bloodstream parasites are taken up, and then they go and they are taken by the flies, and they migrate to the proboscis, where they develop into epimastigotes. So this is a replicated form of the inside the tsetse fly, so they multiplicate. And they undergo an asymmetric division, and one daughter cells stay epimastigote, and another one is a metacyclics, which can migrate back to the mouth part and can be transmitted to the host upon another blood meal from the fly. So once the fly is infected, the fly will remain infective. So one important thing is that at this time it's still not very clear how many metacyclics tsetse fly can transmit to the host, and this is something which I think is really important in terms for vaccine development, because you can. It's really important for to know how many parasites you need to neutralize with the vaccine, but these data are still not very clear. So trypanosomes have developed some way to survive to the host immune system, and one of them is antigenic variation. So antigenic variation is a sequential replacement of the expressed VHG. So VHG is the major protein which is expressed on the surface of the parasite, and the parasite can change sequentially the VHG that is expressed on its surface. And the new VHG will have will be antigenically different from the previous one, which will induce which will render the immune system inefficient to the previous VHG, and another wave of parasites will appear in the blood of the animal. The VHG are also important in the protection of the parasites because there is an estimated 10 to the 7 copy numbers of this protein on the surface of the parasite. So they really act as a physical barrier that prevent the host antibody to access invariant protein, which can be essential for the parasite survival. But some new data have shown that indeed the antibodies can access up to 8 nanometers from the surface of the VHG. So it seems that host antibodies can access some antigen of the VHG. So it might be that the VHG are not forming like an impenetrable barrier like it was previously thought. And another mechanism that the trypanosomes are using to thrive into the bloodstream of the host is that it's an internalization of the antibodies which are attached to the VHG. And they are doing so, so I will show you with this slide. So if you stain the parasite with an antibody directed against the VHG that the parasite is expressing, the parasite can really remove from its surface the antibody by sequestrating the antibodies into the flagellar pocket. So, and you can see that in few seconds, the parasite has completely cleared this antibody from its surface. So that's another important mechanism of protection. And this just shows that the surface of the trypanosome parasite is highly dynamic, and that, yeah, it's that to develop vaccines, you need to take this mechanism into account. So it was some previous attempts for vaccinating animals against animal African trypanosomiasis have been tested. So these are a list of some of them. So you can see that different antigens have been tested, such as flagellar pocket fraction, but also invariant surface glycoprotein, such as ISG65 or ISG75. Some intracellular components have also been tested, such as actin or tubulin. But none of them have shown any very strong protection. And a lot of different protocol has been used, which makes difficult to really compare those results. So for a long time, trypanosomiasis were really thought to be unachievable. But that's something we wanted to address by targeting some invariant cell surface protein for Trypanosoma vivax. So to do so, we used genome-led approach, which is called reverse vaccinology. So for this, we have looked into the genome of the Trypanosoma vivax parasites for selecting genes that are predicted to code for surface proteins. So we design plasmids and synthesize them using gene synthesis. So we took only the parts that code for the ectodomain of the proteins, and we supplant it into an expression vector, which we use to transfect H29360 cells. So we use a mammalian expression system. So we use this system because using mammalian cells increase the chance for the protein to be correctly folded, because essential post-translational modifications are conserved. And then from the 60 plasmids that we designed, we could express 40 recombinant protein in a sufficient yield for testing them into a protection assay using an animal model. I will come back to this in a few slides. So the first thing was to select the genes coding for surface Trypanosoma vivax surface protein, and luckily Andrew Jackson has already characterized the surface of the 3 Trypanosoma species that induce animal African trypanosomiasis, and he has classified the genes into a family, a multi-gene copies families, which are comprising the invariant surface glycoprotein, ISG, or transferrin receptor, or other hypothetical proteins. But we thought that it would be better to look at the genes which have a low paralog numbers to avoid redundant function of these genes. So we just look at low paralog number genes expressed on the surface of Trypanosoma vivax. So we added other criteria. So first criteria is to have a type 1 or GPI encoded protein, as well as secreted protein. So we are sure that they are likely to be accessible by host antibodies. The second criteria was to have a low copy number, a low number of paralogs, sorry. And then we also thought that the protein would have more chance to be accessible by host antibody if the protein can project beyond the VHG coat. So we try to prioritize a protein with a large ectodomain size, and also to prioritize protein that have proof of expression, either transcriptomic or with proteomic evidence. So we ended up with selecting 60 genes with those criteria, and then we express them in the mammalian expression system, and this is the SDS page of all the proteins we have expressed. So you can see that some are well expressed and some other are a bit lower expressed. Some are glycosylated, as you can see, some kind of smear. But we managed to get some transmembrane protein, some type 1, and also some GPI and some secreted protein in our library. So then we needed a mouse infection model of Trypanosoma vivax infection. And we were interested to use the reference strain, the Y486 strain. And we found that we could source one which also expressed the luciferase enzyme. So we can use light-emitted measurement to quantify the amount of parasite into the blood of the mouse. So by using IVIS imaging. So we had two readouts for monitoring the infection of the mice infected with Trypanosoma vivax. So as you can see on the upper part of the slide, you sorry, I should have used the pointer before. We can count directly the parasite in the blood of the mice by using a microscope, but we can also measure the bioluminescence emitted by the parasite. So here in purple, you have a high dose inoculum, and in red, a lower dose inoculum. And you can see that the mice which have been infected with a higher dose develop a parasitemia before the mice infected with a lower dose, and they had to be killed before. And if using the bioluminescence, we could see that you can get bioluminescent signal before the parasite appear can be counted in the blood of the mice. So which is really which makes the bioluminescent reading much more sensitive than the optical counting. And another advantage of using bioluminescence is to increase the throughput of the reading, because it's quite time consuming to count the parasites in the blood of the mice. So for the kind of screen we wanted to do, it seemed essential to have to be able to monitor the infection load using bioluminescence. So now that we have the proteins and the infection model, we started to immunize some mice, and this is a protocol that we have used. So it's quite a classical protocol with a prime immunization where we injected 50 to 100 microgram of the protein associated in combination with alidrogel. 2 weeks later, the mice receive a first boost, and 2 weeks after a second boost. We checked that the mice responded well to the immunization by looking at the antibody titers against the raised against the protein we have used for the immunization. So here you can see that in the x-axis you have all the proteins we have used for immunization, and the mice developed some antibody titer which were different across between the different protein, but seems to be in a sufficient amount. So then we left the mice resting for between 4 to 8 weeks to really avoid non-specific stimulation of the immune system, which could have induced some effect, non-specific effect for the infection. And then we use the Y846 strain to challenge the mice. And we use a challenge dose of 100 parasites by IV injection. And so this is, I also added some details about the infection challenge that we use. So the problem with the Trypanosoma vivax parasites is that the parasites really lose virulence once they are taken out of the blood of the mouse. So to be really sure that we don't have a bias in our vaccine candidate assessment, we infected a cage of control at the beginning of the challenge and another cage of control at the end of the challenge, and we only kept the cohorts of mice where both cages showed a similar level of infection. So this is really to avoid false positive in our screen. And this is the result of the screen that we obtained. So you have here all the 11 cohorts of mice, where for each cohort of mice we have a control group and between 3 to 4 individual proteins tested. So these are the days after infection. And you can see that there is the immunization with individual protein doesn't really induce much difference, except for V8, V31, and V23. In the case of V23, we saw that the immunization with this protein completely blocks the development of the parasite into the mouse. As you can see here, the mouse stayed completely clear from parasites. So this protein is a type 1 protein. It's an hypothetical protein which has no paralog and no autolog with protecongolense. And for a reason that I will just explain later, we decided to call it IFX for invariant flagellum vivax antigen. So after the initial screen, we wanted to confirm that the protein is protective. So we vaccinated a larger cohort of mice. In this case, we vaccinated 15 animals with V23 versus 15 controls. So the control developed the infection and had to be removed from the study at day 8 post-infection, while the mice vaccinated with V23, some of them were still sterilely protected, we had 5 mice which developed delayed infection out of the 15 from the cohort. Then we wanted to investigate the function of this protein, as it's an hypothetical protein. Nothing is really known about it. So the first thing we did is to localize the protein on the surface of the parasite. So I put this slide if you are not really familiar with trypanosome parasites. So in this case, it's a scheme from Trypanosoma brucei parasites. So this is the flagellum of the parasite, and you can see in this transverse view the flagellum with the paraflagellar rod here and the axoneme. And the flagellum is in close contact of the cell body of the parasite and is associated with the fast filament, which is here. And the flagellum is going out of the cell body through the flagellar pocket here. So first, to localize the V23 protein on the surface of the Trypanosoma vivax parasite, we used rabbit V23 antisera and immunofluorescence, and we could see that V23, we could see a signal along the flagellum of the parasite, which is absent when you used naive rabbit sera. And we wanted to confirm this result by using electron microscopy and a monoclonal antibody that we developed, which is directed against V23. And we found that the V23 is localized in patches along the flagellum of the parasites. And the V23 protein can be either found on one side of the flagellum or on two sides of the flagellum in contact in the area where the flagellum is in contact with the cell body of the parasite. So this seems to indicate that the V23 protein is involved in the flagellar function of the parasite, and possibly in the protein is involved in the attachment of the flagellum to the cell body of the parasite. Then we wanted to investigate the mechanism of protection induced by V23 vaccination. So first, we wanted to see if the protection is mediated by antibody, and for doing so, we perform a passive transfer experiment with sera, immune sera to V23. So we vaccinated 50 mice with V23, and we take their sera. And we injected the sera to naive mice, so the day before the infection, the day of the infection, and the day after the infection. And we look if 100 microliter of IFX serum has an effect on the parasite load of the mice compared to control. And we found that the injection of IFX serum significantly reduce the parasite load at day 5 post-infection, and this we obtain a bigger reduction when we injected 200 microliter of V23 immune sera. So this result indicates that V23 induced immunization is, the V23 immunization is mediated by antibodies. So to confirm this result, we generated 6 monoclonal antibodies against V23, and we tested their protected effect using the same protocol than for the than for the passive transfer experiment. And we found that 3 of them could reduce the parasite load of infected mice, so 6B3, 8012, and 3D12, while the other 3 didn't induce any difference. We could see that the protective effect of these 3 antibodies were independent of their binding affinity to the V23 protein, and were not, we couldn't do any correlation of their protection between their protection and the localization of their epitope on the surface of V23, because as you can see, 6B3 and 8012 binds the N-terminal of V23, while 3D12 binds the middle part of the protein. So we isotyped these monoclonal antibodies, and they were all from the IgG1 isotype. And we know that this isotype in mouse, IgG1 antibody can't really activate complement or bind Fc receptors. So we thought that we could change their IgG1 isotype to an IgG2 isotype for the 8012 monoclonal antibody to look if we increase the protection induced by these antibodies. So that's what we tested here. So here it's 8012 IgG1, and we found the same result, that injecting this antibody to the mice can reduce the parasite load. But when we inject the 8012 IgG2 antibody, we really increase the potency of the antibody and completely neutralize the parasites. The gray zone is the background measurement of bioluminescence for the mouse. And we obtain the same pattern for when we inject a bigger dose of antibodies. We then titrate the 8012 IgG2A antibodies, and we found that 3 injections of 20 micrograms of this antibody can induce sterile protection to the mice. So then we wanted to see which function is involved in the protection of this IgG2A antibody, if it's the FC1Q receptor involved in complement recruitment, of the Fc reagent involved in phagocyte recruitment. So we generated monoclonal, we engineered antibody with mutant for antibodies that like the C1q receptor or the Fc receptors, and we injected. It seems that it's not working. I have a problem with the slides. Okay. I will make all of them appear. Okay, so you can see that by injecting the wild type IgG2A antibody, we get sterile protection of the mouse. But when we injected the knockout antibodies for the C1q receptor, we restore we lose the protection. We completely lose the protection, while when we injected knockout antibodies for the Fc receptor, we only partially lose the reception. So this means that complement recruitment is the main mechanism of protection involved in the V23 protection. So we wanted, knowing the mechanism involved in the V23 protection is important and can help us to design the vaccine and to choose the right adjuvant. So we tested different adjuvants which are in use for animal vaccination. So we compared the effect of the V23 vaccination in combination of montanide and Quil A to alidrogel. So we found that montanide did not induce very good protection, but Quil A seemed to induce very good protection. It didn't induce more antibodies or higher antibody titers, but compared to alidrogel, it induced larger amount of IgG2A and IgG2B antibodies compared to alidrogel. So we thought that this adjuvant would be really important for a study, and we confirmed this result by repeating the larger scale immunization experiment. So we have immunized a cohort of 15 mice with Quil A versus 15 control, and in this case we only had one mouse that exhibited a delayed infection, while with alidrogel we had 1/3 of the cohort which exhibited delayed infection. So we really improved the protection by changing the adjuvant for Quil A. So another important characteristic for a good vaccine is that the vaccine protects against a lot of strain of the parasites. So we looked at the polymorphism of the V23 gene across 29 filed isolates, and we found that the IFX gene is really conserved, as we only found 2 SNPs, one non-synonymous SNPs in 2 strains of filed isolates. So this is really not a lot compared to other genes. So we thought that, so apparently IFX is V23 is really conserved genes. We also looked if the V23 protein can be recognized by infected cattle sera. So we tested this into an ELISA using sera from infected animal taking from infected cattle in Cameroon and in Kenya. And we found that the V23 protein is not immunoreactive to infected cattle sera. And this seems to confirm the result from the previous slide, as it seems that V23 protein doesn't seem to be under really immunological pressure, as this would have induced probably duplication of the genes. So another important characteristic of a good vaccine is to induce long-term protection. So that's something we also have tested. So we vaccinated some mice with V23, and we took the sterilely protected mice, and we re-challenged them a second time. And the protected animal, the mice were still protected, and then we re-challenged the mice a third time using a subcutaneous challenge this time, but the mice were still protected. So showing that V23 immunization can induce long-lasting protection. So then, yeah, we wanted to see if V23 immunization could be also protective against T. congolense. So this is the V23 immunized mice challenged with Trypanosoma vivax. And you can see that the mice are protected, but when they are, these V23 immunized mice are challenged with congolense parasite, they exhibit the same parasitemia compared to the control. So V23 V23 immunization does not protect against Trypanosoma congolense, which is something which was predicted, but we wanted to be sure about this. So I don't know if I have time for it, but I wanted to show you some data about the use of the 8012 IgG2A monoclonal antibody as a curative treatment. So we infected some mice with Trypanosoma vivax parasite, and then we injected the 8012 IgG2A monoclonal antibody. So we did 3 injections at day 5, 6, and 7. And then we look at the effect. So we could see that so here you have a development of the parasitemia, and then we treat them with different doses, 50 microgram, 100 microgram, 200 microgram, 300 microgram. And we can see that we have just a delay of the infection course with 50 microgram of the 8012 IgG2A antibody. The mice which received 100 microgram could control the first peak of parasitemia, and the mice with 200 microgram could even control more wave of parasites. Some were completely protected, but the one which received 300 microgram, they were completely sterilely protected. So this means that this antibody can be used as curative treatment. And then we try to refine the dose by injecting only one dose, so either 300 microgram, 400 microgram, or 600 microgram. And we could completely sterilely protect mice with 600 microgram of 8012 IgG2A antibodies. So it's not really to use this monoclonal antibody in the field, but it really allows us to see that you can target an invariant surface protein on the surface of trypanosomes by using antibodies. So to summarize what we have found, so we have identified an invariant vaccine antigen, V23, which is able to induce sterile protection in a mouse model of Trypanosoma vivax infection. So V23 is localized to the parasite flagellum, and V23 induced protection is mainly achieved by complement recruitment. So this V23 seems to be an attractive vaccine candidate for developing a vaccine against trypanosoma infection. And we are now working on translating these results to cattle. And this will need a bit of time because it's quite a big step to go from a mouse to a cow. And we are also interested in using the same approach to try to find some vaccine target against Trypanosoma congolense, and also human trypanosomes such as Trypanosoma brucei and Trypanosoma cruzi. And I will finish by acknowledging my supervisor, Gavin Wright, and also Cécile Cornier, which she has done all the engineering of the monoclonal antibodies. I also would like to say a big thanks to the pathogen lab support, who they have all helped me a lot with the animal experiment, and to David Goulding for the electron microscopy analysis. And I would like to thank you for your attention.